My name is Rukashani Sivalingam. My matrix number is 23DBT21F1049. With me, Ashwini Sivalingam and Sikinu Rafika. I'm here to elaborate about glycolysis. But before that, let me share my infographic. So, what do you think about glycolysis? Glycolysis is splitting of sugar with 6 carbon glucose split into 2 or 3 carbon sugars. These smaller sugars are then oxidized and their atoms are rearranged to form 2 molecules of pyruvate. We have energy investment phase, energy payoff phase. First of all, we're gonna look about energy investment phase. Energy investment phase requires energy endogalactic reaction in the form of ATP. 2 molecules of ATP are used in this phase. Secondly, energy payoff phase. Energy payoff phase, energy yielding phase, exergonic reaction. 4 ATP and 2 NADH are produced. So we have that is energy investment phase. We have 5 steps. First of all, phosphate is ester synthesis, an endothermic reaction catalyzed by exokinase uses energy from ATP to synthesize a phosphate ester to the glucose molecule at the carbon 6 position. Secondly, isomerization. A reaction catalyzed by glucosomerase transforms the glucose 6-phosphate into an isomer fructose 6-phosphate. Number 3, we have phosphate ester synthesis. Phosphate ester synthesis, an endothermic reaction catalyzed by phosphorylation uses energy from ATP to synthesize a phosphate ester to the fructose 6-phosphate molecules at the carbon 1 position. Number 4, the fructose 1,6-diphosphate molecules undergoes a reverse aldol condensation reaction catalyzed by aldolase to form 2 carbon compounds and aldehyde and catenin. Lastly, isomerization. A reaction catalyzed by 3-phosphate isomerase transforms the dihydroxyetone phosphate to glyceraldehyde 3-phosphate. So secondly, we have payoff stage. Payoff stage, we have 5 different stages. First of all, we're going to look at phosphate ester synthesis, an oxidation reaction of the aldehyde catalyzed by glyceraldehyde 3-phosphate leads to the formation diphosphoglycerate. Secondly, hydrolysis of phosphate. A hydrolysis reaction catalyzed by phosphoglycerate kinase forms an acid, a phosphate ion, and ATP. Thirdly, isomerization. The movement of phosphate group catalyzed by phosphoglycerate mutase occurs from position 3 to position 2. Number 4, alcohol dehydration. A dehydration reaction catalyzed by enlase removes the alcohol or negative OH and the negative hydrogen from the carbon 3 and carbon 2. So carbon 2 carbons. Last but not least, phosphate ester hydrolysis. The final hydrolysis reaction catalyzed by pyruvate kinase stabilizes the molecule and forms the acid, a phosphate ion, and ATP. That's all from me. That's all from me. So I'm just passing the next Krebs cycle to Ashwini.
Hello everyone, I'm Ashwini Sivalingam. My matrix number is 23DBT21F1034 from DBT2B. Today I'm going to talk about citric acid cycle, which is known as Krebs cycle. So basically, what is Krebs cycle? Krebs cycle is a cycle of chemical reaction that are the major source of energy in living organism. What is it used for? Krebs cycle used to generate energy through oxidation of acetyl-CoA, and it is used for synthesis of NADH and also production of amino acid. Where does it happen? Krebs cycle happens in mitochondria for eukaryotes and cytosol for prokaryotes. Let's get into the cycle. Okay. It begins with a preparatory step, which is pyruvate. It is derived through the glycolysis with 2 carbon, which we just saw just now. The next step is the oxidation of pyruvate into acetyl-CoA by the enzyme pyruvate dehydrogenase. In this reaction, a molecule of carbon dioxide, NADH, and hydrogen produced or generated. For the next step, acetyl-CoA combines with oxaloacetate to form citrate. In this reaction, citrate synthase works as the catalyst. Next, as I can make it short, citrate is isomerized into isocitrate by the enzyme aconitase. Then, isocitrate is oxidized to alpha-ketoglutarate by the enzyme isocitrate dehydrogenase, and a molecule of CO2 is generated here. After that, the next step is a molecule of NADH is reduced to NADH in this reaction too. After that, the next step is alpha-ketoglutarate converted into succinyl-CoA. This process by the enzyme alpha-ketoglutarate dehydrogenase. In this reaction, a molecule of NAD is reduced to NADH and a molecule of CO2 is released. The next, succinate is converted. The next is succinyl-CoA is converted into succinate by the enzyme succinyl-CoA synthase. In this reaction, a molecule of GTP produced. After that, the next process is succinate is converted into fumarate by the enzyme succinate dehydrogenases. After that, this fumarate will be converted into malate by the enzyme fumarase. And after that, as the final step, malate is converted to the oxaloacetate by the enzyme malate. Malate dehydrogenase. Here, NAD is reduced to NADH. That is the simple way to understand citric acid cycle. As the result, 2 molecules of carbon, 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of ATP or GTP is produced. This is for a single carbon molecule. We need to know that after glycolysis, the pyruvate will be 2 carbon, which means all these molecules will times into 2 as how 4 molecules of carbon, 6 molecules of NADH, 2 molecules of FADH2 and 2 molecules of ATP or GTP will be produced. That's all from me. Thank you.
Okay. Hi and Assalamualaikum. My name is Siti Rafika Nasir. I'm from DBT2B. Now my turn to describe or present about the electron transport chain. That means the ETC, the short form. Okay, now I want to introduce about the ETC or electron transport chain. Okay, the first, a very little energy is produced during glycolysis and kreb cycle. The second is actually this is not located but the locked LOCKED. So my fault, sorry. Locked in the original glucose released by the electron transport chain and chemiosmosis. The third is the ETC is the network of the electron carrying proteins. And then the fourth, the energy rich molecules because contents of the electron with high transfer potential. And then the fifth, used to reduce molecular oxygen to water. Free energy is the liberated used to generate ATP. And beside here, this is the electron carriers in the mitochondrial inner membrane transfer electrons from NADH and FADH2 to oxygen. The point is the first, the 3 proton pumps that are located in 3 of 4 electron transport complexes. The second is the energy released during electron transport. The third is the protons cannot diffuse back into the matrix. The fourth, the generating ATP and ADP and inorganic phosphate. And the fifth, phosphatic group. Okay, the next, this is the during electron transport along the chain, electron carriers the alternate between reduced and oxidized states as they accept and donate electrons. The point is the reduced when it accepts electrons from its uphill neighbors, lower affinity for electrons. The second is the oxidized from its passes the electrons to its downhill. And the third, a favor protein. It has a phosphatic group called it has a prosthetic group called flavin mononucleotide. The simple word is this FMN, the short form, sorry. And then the next point is the passes the electrons to an iron sulfur protein. Then the call UBQ9. The short form is Q or COQ. This is the iron sulfur protein. Then passes the electrons to compound called UBQ9. Sorry. And then electron carrier is a small hydrophobic molecule. And then protein called cytochromes. And then the next point is the prosthetic group called a heme group has an iron atom. That means the diaprosthetic group called a heme group has an iron atom that accepts and donate electron. And then the electron transport chain has several types of the cytochromes. And then the source of the electrons chain is the FADH2. And the last, the FADH adds its electrons transport chain complex 2. The next part is this is the chemiosmosis, the couples the electrons transport chain to ATP synthesis. The point is the NADH and FADH2, the shuttle high energy electrons. The second point is the arrows trace the transport of the electrons. The third point is as the complex 1, 3, and 4 the accept. And the last point is the oxidative phosphorylation. And then the last part, this is the energy from the proteins, carbohydrate, and fats. So thank you for listening my presentation.